Hello, everyone. Thank you for taking time out of your day to attend this lecture in neuro-oncology. This lecture is being recorded during the time of the COVID-19 pandemic, so I do wish and I do hope everyone is safe and well and healthy. To those that have been directly affected by the pandemic, I wish you great strength moving forward. I'm sure as humans, regardless of the climate we're in, we will be able to find resolve. My name is Gebik Safaryans, and I am currently a Master's of Science student in UCLA's Department of Bioengineering, specializing in the neural engineering subtrack. And I will be leading today's lecture on how a systems thinking approach can make way for the development of new therapeutics for a very specific and detrimental form of central nervous system cancer called glioblastoma multiforme. Now with that, I will shift to the slides. Before I begin, I would like to thank Dr. Carlos Cepeda for allowing me to lead this lecture. I was a student in his class in the spring of 2019, which was my last quarter at UCLA undergrad, and I found it quite productive and thought-provoking to engage with the guest lecturers from all diverse fields. Now, hopefully I can serve my purpose in helping you understand the current climate of research behind glioblastoma therapies. And should you have further questions or interests, I do encourage you to engage me via email at skevik@ucla.edu. During this lecture, I must emphasize that at the end of the day, regardless how much microbiology or engineering approaches to therapies will be discussed, it's imperative to maintain the affected patients and families at the center of thought in developing any new systems for whether it be diagnosis, treatment, beyond. And as we'll see, symptom onset and progression in patients is quite swift and alarming, causing a great deal of burden to who may be mothers, fathers, children, aunts, husbands. To push the boundaries, as they say, it's important to keep people at the center of thought. Next, we'll discuss the shortcomings associated with the current standard of care model for treating glioblastoma, or GBM, which I'll refer as for the remainder of this lecture. While many FDA-approved drugs continue to be instated, the standard of care has become a sort of dogma. And finally, to push the boundaries again beyond what is the standard model of care now, we must change our perspectives. So the idea that drugs work well in a traditional cell culture and are just immediately translatable to in vivo physiological systems is now beginning to be considered primitive. Through this lecture, hopefully you will discover new approaches to model glioblastoma response to newly developed treatments and understand exactly how those systems in the extracellular environment may deter response or mount a resistance, as we say, to these treatments that are considered standard, thereby actually worsening patient outcomes. Before beginning our discussion about glioblastoma, or GBM more specifically, let's take a step back and talk briefly about the field of neuro-oncology in general. So onco, the root, comes from the Greek word onko, meaning burden or a mass. So neuro-oncology quite literally translates to the study of these burdens or masses present within the nervous system. Now, formally, the American Academy of Neurology defines neuro-oncology as a specialty involving the management of both primary and metastatic central and peripheral nervous system neoplasms. Now, the term neoplasm, a neoplasm is an unregulated growth in tissue, as we can see in the diagram below. So it can either cause harm on the neighboring tissue and be labeled as malignant, or it can be neutral and have benign effects, as they say. So that's the differentiation between a malignant and benign cancer. Cells regularly do proliferate and renew. Not so much, of course, in the central nervous system, but there are specific hotspots. And under normal circumstances, the amount of proliferation is tightly controlled by locally secreted factors. So cells communicate with one another to halt this excessive tissue growth that's seen in neoplasms. When the system of wide communication is disrupted, of course, cells begin to indefinitely multiply and form these large neoplasms. And as you can see in the image above, these brain tumors can become quite substantial, and they may spread to cover the course of multiple lobes, contributing to multiple function deficits presented in those patients that are affected. And the mass visibly is contributing to this increased intracranial pressure, so physically pushing and narrowing what is in this case the body of the lateral ventricle and causing local cellular damage. And of course, beyond leaving a scar, it's quite visible in the brain, it leaves a scar in the minds and in the skins of these patients. And the image below is of a nurse who underwent a resection of a grade 3 astrocytoma. So they took out the tumor. And unfortunately, many people, due to this experience, even following this resection, even following this invasive procedure, experience depression as a comorbidity. That gives us more room and more of a reason to push the boundaries for patient recovery from this high-grade glioma, that is glioblastoma, as we'll see. As further background, about 30,000 newly diagnosed cases of central nervous system neoplasm in general, so not just glioblastoma, are projected to occur just in the year 2020 alone. So as the 10th leading cause of death in adults, both male and female, it's typically presented in the cohort aged about 55 or older. I've seen some reports say 64 being the median age, and it's predominantly seen in males. So rarely in children, and more specifically those under the age of 15, they may also experience this tumor growth, but again, very rare. And a majority of central nervous system tumors localized to the cortex are presented primarily in the frontal or in the temporal or parietal lobes at 26, 19, and 12% specifically. But rarely neoplasms, of course, you will also see them present themselves in the occipital lobe, even the brainstem, or even the cerebellum. But classifying these tumors can be done in many ways, not just where they're localized. One of these ways is actually based on their place of origin. So actually a majority of these central nervous system tumors are metastatic in that they travel from separate organs outside of the central nervous system, meaning mainly the lungs, actually the kidneys, and even the breast. They travel through the bloodstream and into the brain or spinal cord. Now, otherwise, if they're not metastatic, and then they're mainly considered primary tumors, meaning they develop, of course, from the native brain itself. So they're not a result of another form of cancer traveling to the brain and spreading there. But most importantly, of course, is the measure severity. And this is the most important measure when it comes to determining patients' prognosis. So whether they are in critical conditions of health or not. So those with a grade 4 neoplasm are considered to be in critical conditions of health, and very few actually survive beyond 5 years following a formal diagnosis, of course. Those with grade 1 tumors tend to resolve, thankfully, and may not exhibit any alarming or clear symptoms to receive a formal diagnosis at all. This is comparable to cysts, right? So packets of fluid that fill up the brain and actually go unnoticed throughout life, and they're only detected following a postmortem analysis. Now, the World Health Organization has designated a grading scale in order to uphold uniformity in diagnosis and the further classification of each tumor type. Now, those listed in the table are only a handful of the many forms present across patient populations. Pilocytic astrocytoma, ganglioglioma, pineocytoma, they're all forms of relatively low-grade tumors, meaning they exhibit some quality of slow growth, lower likelihood of spread, right? And they're often candidates for surgical resection. Grade 2 tumors are more likely to return even following the surgical intervention. So that's what makes them different from grade 1 tumors. Those still relatively benign, many of which, of course, can be malignant. But more high-grade neoplasms, particularly, they're very difficult to treat. And they may maintain a population of these rapidly dividing cells, again, the proliferation, as mentioned earlier, angiogenesis, or the formation of new blood vessels, and quite rapid metastasis, actually, throughout the brain. So glioblastoma multiforme is actually the most lethal of all high-grade gliomas, and will actually be the focus of the rest of this lecture. As can be told from the name itself, glioblastoma is glial in origin. It is a form of glioma. More specifically, it is an astrocytoma, meaning astrocytoma in origin. And of all primary tumor types, this is the most common, aggressive, and lethal form. Again, I would like to emphasize this. It affects less than 10 out of every 100,000 persons. Of course, some estimates I've seen go as low as 2 or 3 out of every 100,000 people, but it makes up about 80% of these malignant brain neoplasms and 15% of central nervous system tumors in general. And it typically affects those over the age of 55, right, as with most central nervous system tumors. And the highest incidence is seen in those specifically in the 55 to 60-year-old age group. Now, I want to make this clear. Although the disease is considered rare in the general scheme of all disease states possible, of course, this does not mean that we simply neglect its presence, right? Even if it affects just one person, that one person deserves our full attention and effort to a better quality of life. By simply taking this account from one Christopher Stewart Elliott, we can begin to realize the uncertainties and the troubles associated with the patient side of symptom onset and disease progression. Now I'll read his quote. He said, "All of a sudden, I felt nauseous and I kept smelling the most awful smell. A very strong smell that smelled like very old sneakers." Although this isn't any dead giveaway of a brain tumor, of course, Christopher you can see, is beginning to feel and realize this system-wide disturbance, smell distortion, internal distress. Later, he experienced seizure onset, each of which are a consequence of this quarter-sized brain tumor, increasing intracranial pressure, contributing to the changes in the activation patterns in neurons responsible for smell. This further increases the likelihood of seizure onset, which we saw in the case of Christopher, unfortunately, especially by disrupting these many neural networks in the temporal cortex, where his tumor resided. Now, keeping optimistic, Christopher had still not grasped the magnitude of the situation at hand, even following the first resection of the tumor. Believing his life was to return to normal. He said, "I would have surgery to remove the tumor. Okay, sounds simple enough. My thinking was that, all right, we'll have the surgery, recover, and then life will go on and all will be fine." And no matter how much we wish this was so, relapse and infiltration of even primary brain tumors to deeper regions in cortex results in further complication for these patients. As was the case with the second tumor, nothing could be done, and Christopher was actually only given 3 months more to live. And eventually passing away, Christopher remained hopeful for the future of glioblastoma care. And at least we as a field should have patients like Christopher rest assured that following an initial resection, the chance of secondary tumor progression is little to none. That should be our goal as a field, beyond preventative care. But clearly, there's still much work to be done, as we'll see. Even now, unless we have MRIs, PTs, CTs as a part of your regular checkup appointment, detection of glioblastoma goes actually quite undetected. Now, cases of early detection may occur, of course, some by chance. Nonetheless, the symptoms listed here in headaches, combined with seizures or changes in mood, definitely shouldn't go unaddressed, especially if you're a caregiver or a peer of someone who is expressing these symptoms, and they're quite actually easy to detect, right? Changes in attitude in the person, these should immediately be reported, especially if they're quite substantial. But after all, these symptoms are global, meaning the same symptoms can be generally experienced across different tumor manifestations in patients. And these transient symptoms experienced can lead to actually different differential diagnoses. So a full scan, of course, is necessary for proper detection of this cancer. A true diagnosis of glioblastoma can be given following patient biopsy. So the first hallmark observed in the resected tissue, the tissue that is removed for biopsy, are the presence of this microvascular proliferation, or MVP, meaning there's presence of endothelial cells, and if not just the cells, fully matured microvessels penetrating the tumor mass. Now, GBM cells have this increased tendency to associate with local vasculature within their pericellular niche in general. And there are reports of stem-like glioblastoma cells differentiating into the endothelial cells that give rise to this vasculogenesis, angiogenesis, contributing to this proliferative phenotype. Now, a second important biological and diagnostic factor is this presence of the pseudopalisading necrotic core as a result of hypoxia within the core of the tumor itself. So if you look within the core of this tumor, you will see cell death. And specifically at the center is this presence of glial-like stem cells at high density, along with cell death. And this region is hypoxic relative to its surrounding periphery, meaning the core is, of course, subject to poor oxygenation. And the immediate periphery of this core actually exhibits a higher rate of proliferation and migration due to the presence of these microvessels, as mentioned earlier, physically feeding and supplying nutrients to the constantly migrating and self-renewing cell populations. The core and the periphery are the areas that actually end up putting the most defense. So they contribute to the most resistance to applied chemotherapies. And as we'll see, it's quite multifaceted in nature. Now, regarding a means of care, patients typically undergo a standard 3-part procedure, beginning with resection. And depending on patient status, the burden as a result of the tumor and, of course, the reachability, different degrees of residual may remain following this resection. Now, residual refers to the amount of tumor remaining within the brain following a successful resection. A small portion of the removed tissue is then used for the biopsy, as spoken about in the previous slide. That's to deliver primarily more formal diagnosis. And there are different types of residual that occur. So a gross total removal occurs when the entire GBM mass is removed and very few cells remain within the site of resection. Normally, following a successful resection, patients undergo rounds of gamma radiation therapy. Now, should the tumor actually be inaccessible, relatively low in mass, or should it be benign, or actually even should the patient refuse invasive intervention, because of course it's up to the patient, then they can skip directly to the second stage. And they can receive this non-invasive gamma radiation procedure known as gamma knife radiosurgery. However, in the case of most glioblastoma patients, of course, they should regularly undergo resection due to the just sheer invasiveness and the progression of the tumor itself. And it's, of course, resection has been shown to increase the quality of life and strictly the lifespan of our patients. Now, mechanistically, what does radiation do? Radiation, once you've successfully completed this resection procedure, perturbs the DNA integrity of those tumor cells, and that leads to cell death, or as it should. Now, localizing treatment to the tumor site further increases the chances of removing even these miniature tumor cells, these residual cells left following the initial resection procedure. Then, of course, this is combined with chemotherapy treatment, as we see with most cancers. And it's with an agent called temozolomide. And typically, it's 5 days on and 23 days off, full 28-day cycle. And it's repeated at least 6 times And with an upper bound, of course, 12 cycles, as we see, as many patients tend to seep further into worse condition, worse status near the end of their life. It's typically seen up to 12 times before a patient passes. And with temozolomide, the median GBM survival has actually increased by about 2.5 months. More and more patients are seen to survive at least 2 or even up to 5 years. While any feat against a lethal cancer like GBM is excellent and we should surely celebrate, there remains far more work to be done. 2 months in the grand scheme of things, in the grand scheme of life, after all, is actually quite insubstantial, especially when patients are weak as a result of chemotherapy administration towards the end of their life. So to progress, it's important to keep in mind that we need to first formulate a better understanding of exactly how temozolomide takes its effects on GBM cells, and this was actually done. Temozolomide itself is this well-characterized alkylating agent, meaning it attaches an alkyl group, specifically a methyl group, to DNA nucleotide, purines or pyrimidines. Now, when this occurs at a high scale, cells are unable to harness their innate DNA repair mechanisms. Cells are equipped with DNA repair mechanisms, of course, when DNA undergoes damage. As in the case with temozolomide, it's methylation. And the cells become overwhelmed, and when there's too much alkylation happening, they undergo apoptosis. So when the room for repair is not substantial, the cells should go under apoptosis or cell death. Now, while that's how things should work, what you'll find is things don't just translate perfectly into the real world from the laboratory, of course. There are nuances and critical features about a system that cannot be fully represented in an experimental context. And this introduces great bias in findings. So as it pertains to temozolomide specifically, cells are equipped with DNA repair mechanisms, as mentioned. And should a cancer cell's DNA not become overwhelmed by the methylation of temozolomide, it initiates the transcription and translation of this enzyme known as O6-MGMT, or O6-methylguanine methyltransferase. And this enzyme cleaves the methyl group right off of the affected DNA base pairs in the active site. In the active site of this enzyme is actually a cysteine group that accomplishes this through its diol functional group. So it restores the DNA and maintains its proper function, keeping the cancer cell actually alive and intact. So this is the resistance mechanism against temozolomide. And when this was known, of course, immediately an agent targeting this resistance mechanism was created. And while clinical trials were performed, it was found that actually inhibiting O6-MGMT, it does promote increased life by about 2 months. Again, great, but that's nowhere near the end of the tunnel. So if targeting the repair mechanisms doesn't actually work, and targeting this specific repair mechanism specifically doesn't work that well, then what else is there? This is the question we actually continue to ask as a field. Now, what else beyond intracellular genetic or epigenetic factors can we target in the microenvironment to improve therapeutic outcomes for our patients? We need to broaden our perspective. We need to broaden our perspective when it comes to understanding this disease, and we must adopt a systems thinking approach from here on out. That is the only way to answer the question. Now, rather than limiting ourselves to studying cells in isolation, as scientists and as thinkers, we must be able to develop systems that compartmentalize each component contributing to glioblastoma phenotypes, whether it be migration, proliferation, prolonged survival. These are all what contribute to the observed resistance and, consequently, low quality of life for our patients. In this lecture, we'll explore the 3 realms or components shown here, which must be addressed, of course, in developing future solutions. And those are the immune system, blood-brain barrier, and the extracellular matrix. To begin with the blood-brain barrier is essential. Acting primarily as a filter, it's selectively permeable to a lot of small molecules and compounds that are exposed to local neurons microglia, and other glial cells. Differing from the regular blood vessels found in the periphery, these vessels are lined with parasites and astrocytic end feet that actually further reduce the efflux of unnecessary foreign or even possibly toxic chemicals to the local cells in the central nervous system. Now, this has drawbacks, of course, in terms of administering medications, especially temozolomide. That's quite impermeable to this blood-brain barrier. Now, to improve drug delivery means taking advantage of already present regulatory proteins lining the barrier, as we'll discuss, evading these filter proteins completely, or developing novel systems that do not rely on the blood-brain barrier penetration for delivering chemotherapies. These are the three things that will be discussed in developing new therapies going against the blood-brain barrier. Now, the barrier is saturated with substrate-specific transporters for a variety of molecules, including glucose, methionine, or histidine amino acids, insulin, and ketones. Essentially, key molecules for anabolic and catabolic processes that are required for cell respiration and survival. Now, while it can be possible through a means of an adeno-associated virus or other virus subtype to instill these gene targets coding for chemotherapy-specific transporters to be expressed in these blood-brain barrier cells, it's quite difficult to translate clinically, and such viruses are still not well characterized in terms of their potency to induce an immune response in patients. But nonetheless, of course, there are more practical methods, and one is taking already existing chemotherapies and physically conjugating them or attaching them chemically to peptides that are already being transported across the blood-brain barrier. So essentially hijacking the system. In a study by Ying Li and colleagues, the group targeted the lipoprotein-related receptor protein 1, indicated here as LRP1. It transports angiopep2 into the extracellular space. Angiopep2 is a specific peptide traveling through the blood vessel. Again, that's just one of the transporters, right? There are many. So what this group did is they conjugated 3 paclitaxel molecules. Paclitaxel is a form of chemotherapy, to a single angiopep2 molecule. And they actually observed a 50-fold increase in drug delivery to the tumor site. Now, again, paclitaxel is a chemotherapy against glioma specifically. And they conjugated to the substrate and called this in unison ANG0015. And it actually completed phase 2 clinical trial, and it's still yet to complete phase 3. But this just serves as one example I wanted to show everyone of how you can actually hijack the system, right? There are many transporters for glucose, amino acids, insulin, ketone, like I mentioned earlier. So we can utilize these compounds to sort of conquer this barrier. Temozolomide can be conjugated to actually any of these as a means of improving delivery into the extracellular space. The blood-brain barrier is also coated with drug efflux proteins, and they potentiate this chemotherapeutic resistance response. P-glycoprotein and breast cancer resistance proteins, or BCRPs, they're generally classified as ATP binding cassette transporters, so they're separate from the transporters I mentioned earlier. And they generally inhibit any extraneous access to the pericellular niche, right, the tumor niche. Now, as demonstrated in the image below, they are seated generally towards the open lumen, that is the blood vessel, thereby preventing drugs to even penetrate beyond the point of what are endothelial cells lining the blood vessel. Now, in vivo experiments, and some have demonstrated, and I've shown here, tumor growth can be inhibited at a higher degree by blocking either these P-glycoprotein or BCRP receptors. And in one 2018 study published in the Neoplasia journal, actually, the experimenters either genetically knocked down the 2 receptor types or administered alacridar as this potent receptor blocker for ATP binding cassette transporters. And upon co-administration of temozolomide, they saw increased survival in their mice by actually 8 days and significantly reduced tumor size. compared to those mice that just received temozolomide alone. So while genetic modification of the blood-brain barrier to express these knockdown genes is difficult to achieve, again, difficult to translate clinically, alacridar is one example of a drug species that can be developed that can be supplement to temozolomide or other existing chemotherapies to increase its efficacy. And this is actually quite comparable to what's done in Parkinson's disease, right? Parkinson's disease, we have levodopa. But what's administered with levodopa to increase its effect on the central nervous system is carbidopa. A third mode of delivery is actually a form of cheating the blood-brain barrier. I like to say you cheat it because you completely bypass the system itself. Now, these gels, which are made of biocompatible chemicals, polymers, it was done in these two studies and administered both in vitro and in vivo. In vitro as a hydrogel and in vivo as the oncogel in the study below. Now, these gels can be seeded with an appropriate density of chemotherapy, and they can actually be injected to the tumor site. So within the body, the biodegradable gel will slowly disintegrate, releasing these drugs, these chemotherapeutic drugs, at a controlled rate, at a known rate, according to the degradation properties in vivo. So in the top figure, gemcitabine, a chemotherapy agent, was administered either in isolation or in a hydrogel subcutaneously adjacent to U87Mg tumor xenograft in mice. Now, clearly the hydrogel made a difference. The hydrogel provides a sustained delivery model, and that means the gel can be dosed to provide sustained release over a long period of time. And again, to help you gain a better understanding, gels are actually the structures that result from cross-linking between different materials like collagen or other polymers. And they have this known degradation rate. So while they're under the stress of pH or temperature or other local cues, when administered near a tumor, right, in the body, we know this degradation constant, meaning we know how these gels are going to degrade and deliver chemotherapy locally. So we can dose these gels with a specific amount of chemotherapy and know its degradation rate and delivery rate locally. So more than this, in a separate study, of course, that's shown below, this oncogel was created. And when it was administered with TMZ or temozolomide and radiation, this resulted in actually substantially long survival in rats. And even oncogel with radiation alone in the absence of temozolomide treatment was actually very substantial. Now, gels provide this advantage, meaning they can be created with biocompatible materials that don't induce any sort of immune response against the gels themselves. Now, they have controlled degradability rates, of course, as I mentioned, and they can be released locally rather than systemically. So chemotherapies are typically, they cause systemic damage, and that's the result of being infused into vasculature, and either orally or intravenously. But this new mode, this use of gels as a delivery may actually dampen the systemic deterioration that's actually associated with intravenous delivery. And it can even further potentiate its response. So you have less of a response systemically, meaning you improve patients' quality of life. So this is actually a great field, and it has great potential in the near future. And I'm actually very excited to see different gels, not only in glioblastoma, as we'll see with many different disease states. They're developing gels to deal with drug delivery for chronic pain, migraines, the list goes on. So very exciting field. Now, glioblastoma itself is innately immunosuppressive. So this further contributes to the mounted immune response. And while I can't give an in-depth review of immunology, there's no use. I'm going to be introducing just a few topics so I can give you a better understanding of exactly how T cells specifically, T cells are a specific type of immune cell, they can actually play a harmful role. Now, future therapies, of course, have to develop on, have to focus on actually limiting the harmful interactions between these glioma and these T cells. And even as you'll see in the image here provided, these tumor-associated macrophages. T cells are the foundation of a mounted attack against an immunogen. Any agent that evokes an immune response is known as an immunogen. Again, I'll be defining terms that may be foreign to some of you. Now, how T cells normally recognize whether agents are foreign or native to our body is by first binding T cell receptors, TCRs, and shown here in blue, and these major histocompatibility complex receptors. And they're presented in actually all cells of our body that include a nucleus. Now, besides this one bond that forms here, there's actually a second bond that's very important to determining attack versus a suppressed response by these T cells. Now, certain proteins can be co-stimulatory, and upon binding T cells, they activate immune response against the antigen. So these are known as co-stimulatory. Ideally, should this be the case in glioblastoma tumors, we would see an avid immune response neutralizing the cancer cells in their place. So there should be no GBM compression. But of course, this isn't the case. And glioblastoma is actually quite tricky, for lack of a better word, in that it downregulates these MHC receptors, and meaning the recognition, of course, the binding of T cells cannot occur there. And if not just that alone, it expresses actually co-inhibitory rather than co-stimulatory cell proteins like PDL1, CTLA4, some not shown in the image, TIM3, TIGIT, and they either bind local macrophages or T cells and elicit this immunosuppressive response, meaning it accomplishes the opposite goal of co-stimulatory proteins, right, that normally initiate an attack. These are co-inhibitory proteins. Now, this is also complemented by, and this is found in many papers, there's a recruitment of T regulatory cells into the tumor microenvironment. And T regulatory cells actually further cause immunosuppression, further dampen this T cell response from taking place. And over the years, many companies actually have produced many monoclonal antibodies that actually target these PDL1 or CTLA4 receptors to inhibit immunosuppression. While in isolation, many of these treatments have actually shown some success, future stage 2 or 3 clinical trials have to further evaluate whether they are truly beneficial when given in place of temozolomide as a standard therapy or in conjunction with temozolomide. So as more immunosuppressive receptor targets are discovered, and I'm sure many more will be discovered, there's definitely going to be more antibodies specifically targeted to different ones. Besides antibodies, there came the advent of CAR T cell therapies, or chimeric antigen receptor T cell therapies. Now, before explaining how this works, I want to make sure everyone has a formal understanding again of how T cell recognition works and how T cell attack occurs. Now, T cell receptors contain these extracellular subunits that actually bind to MHCs. Now, in humans, MHCs specifically are termed HLAs, and this term may seem familiar to you if you're familiar with blood typing. Now, they also have this intracellular CD3 binding domain that participates in activating an immune response should the T cell form, of course, another protein-protein interaction with another set of co-stimulatory proteins. Unfortunately, not shown in this image here. What needs to, of course, occur is one, this interaction, right, and CD3 activation, and another set of co-stimulatory protein interactions for a full immune attack to be launched against the target cell, which is in this case a cancer, glioblastoma. Now, as I mentioned earlier, these glioblastoma cells are quite tricky. And what they do is instead of activating their co-stimulatory cell expression, co-stimulatory protein expression, they activate co-inhibitory protein expression in the membrane, which means, yes, this binding may occur, right? Meaning even if these HLAs are present, but it's likely that a co-inhibitory protein association occurs that actually dampens immune response, that prevents this T cell attack from taking place. Now, this is where CAR T cells can actually serve as a benefit, as we'll see. Now, autologous T cells, meaning T cells derived from the patient, can be collected and precipitated. Gene targets specifically coding for chimeric antigen receptors can then be instilled in these cells and delivered back into the patient's circulation. Now, these chimeric receptors bind to specific antigens presented by tumor cells extracellularly. And upon binding, intracellular co-stimulatory domain and CD3 domains both activate. Now, looking at the importance of this is quite substantial. So to understand this, look, we can design these chimeric antigen receptor extracellular units and prime them with these specific tumor-specific antigens that can bind to receptors along the tumor cell. And these receptors are actually only found upregulated on tumor cells, so you're likely to target them with these T cells. Now, upon binding, what you see is there's no longer the need for this dual receptor interaction to occur for co-stimulatory response to take place. It's all done on this one T cell subunit here shown. The chimeric antigen receptor has both the co-stimulatory domain that activates along with the CD3 domain that activates, meaning 2 becomes 1. Now, the extracellular chimeric receptor region, it can be primed again with many tumor-specific antigen targets. And many upregulated receptors that have been found in tumor cells in these glioblastoma cells include EGF receptor, HER2, and IL-13RA2 receptor. They can all be used to prime CAR T cells. And actually, in a recent paper by Wang and colleagues, they identified actually one specific tumor antigen as chlorotoxin. And it's a peptide derived mainly from scorpion venom. And it can serve as this potent GBM targeting ligand because it has a weak association with any other cell found systemically, mainly targeting GBM cells. But the receptor that it targets is actually still unknown. But of course, what's important here is this opens the door for expanding our CAR T cell antigen repertoire, the antigen of these chimeric antigen receptors. Now, that allows us to create more composite therapies to target multiple receptors simultaneously. So ideally, you can see in the future, maybe there's a medication round. Instead of the current temozolomide standard of care cycle we see now, there's a medication round targeting EGF receptors, HER2 or IL-13RA2 receptors, and the hopefully then known chlorotoxin receptor, right? That may be a very valuable realm to explore. Beyond cell-cell interactions, there's a whole bounty of extracellular matrix proteins and polysaccharides that also contribute to this mounted resistance response against what is known as the current standard of care. And this calls for the development of new chemotherapies that instead of targeting the DNA repair mechanisms, as we saw with the O6-methylguanine methyltransferase enzyme, those that target specific cell to extracellular matrix or ECM receptors and their associated membrane protein targets. One extracellular glycosaminoglycan, that's known as hyaluronic acid, or hyaluronin, is presented in a mixed distribution of low molecular mass and high molecular mass forms throughout this tumorigenic niche. Now, most notably, it is present in a higher concentration surrounding vasculature, as we saw with previous diagnostic factor being angiogenesis and tumor cells associating near vasculature. And that's where most GBM cells, again, proliferate and reside. Now, these large polymers, hyaluronic acid or HA, they bind to either CD44 or RAM receptors, RAM being receptor for hyaluronic acid mediated motility. And they actually saturate the glioblastoma membrane. And both CD44 and RAM are associated with increased cell motility and proliferation, not only in glioblastoma cells, but it's also been studied and observed in breast and ovarian cancer models. Many of these axes, actually, intracellular cascade axes, are found there too, outside of glioblastoma. So the ERK and the RAS and RAF axis are actually believed to be activated downstream factors that are responsible for what you'll see here down below, cytoskeletal rearrangement. And of course, these are all leading to gene targets that lead to cytoskeletal rearrangement. And this actually leads to the observed cell motility and the consequent invasion phenotype we see with these cancer cells. And of course, these intracellular cascades are believed to be the foundation of tumor metastasis itself. So in fact, it's actually been shown that patients with higher CD44 saturation in their GBM cells actually had lower survival rates, 3.5 months compared to 18.5 months. Now, this, of course, is after biopsy or post-mortem analysis. The significance of each receptor to the glioblastoma cell subtype is actually yet to be addressed. So the contribution of either RAM or CD44 or both together, the significance of them is yet to be determined in the different types of glioblastoma cells that are present. So another thing you'll actually find is there are many different types of glioblastoma cells, right, based on their mutations that are present. So it's not all one form of cancer. There are many different forms of mutations that contribute to glioblastoma phenotypes. So some actually are mesenchymal, they have higher motility, classical or the proneural classifications of glioblastoma cells. So there are different subtypes, of course. And it's actually yet to be determined exactly for which subtype, how much CD44 or RAM individually contribute or how much they contribute in unison. So knowing this can actually give us tools for developing more effective strategies that target specifically CD44 or RAM after we do tissue typing. So after we know what type of GBM tumor is presented within our patients. So besides hyaluronic acid, there are also many glycoproteins that are also present within the extracellular matrix. And numerous papers have demonstrated that the upregulation of factors like tenascin C, vitronectin or fibronectin within the extracellular matrix itself are actually present in higher grade gliomas. And cells themselves actually deposit their own glioblastoma protein. Besides hyaluronic acid, there are many glycoproteins that also are present within this extracellular matrix. And there are actually numerous papers that have demonstrated that an upregulation in what may be tenascin C, vitronectin, or fibronectin, all glycoproteins within the extracellular matrix itself, that's actually very prevalent in higher grade gliomas. Now, what's alarming is actually the cells themselves, right, as any normal cell, they deposit their own glycoproteins into the matrix itself. And as you can see, that can actually contribute to this feedback loop, this positive feedback loop in terms of a response. Because if these factors are contributing to more and more proliferation or migration, just depositing more, you're going to see an increase in that, obviously. And many of these glycoproteins actually, those that aren't even mentioned here in this slide, they actually contain a binding unit specific to receptors along the glioblastoma cells. These receptors are known as integrins. And these integrins bind to specifically the RGD sequence, amino acid sequence, or the arginine, glycine, and aspartic acid amino acid sequences that are served as, of course, these binding units. Now, this contributes to this activation of a similar group of cascade proteins, as we saw, of course, from the previous slide. We saw a cascade of proteins being activated immediately after membrane ECM cell interaction. We saw RAF, here we're seeing PI3K, AKT, ERK, but beyond the secondary signal activation, integrins actually physically associate with actin filaments intracellularly, and these actin filaments are actually what contribute to cell motility, meaning that there may be this direct role of glycoprotein binding and cell motility that even may forego many of these intracellular factors becoming activated, many of these secondary responses becoming activated. It could be quite direct. Now, taking into account these glycoproteins is actually quite important because it's not like they're cell-secreted factors. Well, it's not like they're cell-secreted factors in the meaning that they're not specific proteins or small molecules secreted by cells that then go on and infiltrate nearby cells and contribute to this genetic response. But in fact, they're just things that contribute to matrix formation, right? And by way of the formation of the matrix, based on what components are present in the matrix, these integrin-specific interactions can take place and can actually lead to a different phenotype observed in these glioblastoma cells themselves. Now, integrins, growth factor receptors, these are all future targets for therapies. And they've been targets of actually many new chemotherapies that are in market that specifically target these EGF receptors or these FA3 receptors, VEGF, vascular endothelial growth factor receptors, each of which may actually be integral to this integrin-mediated response. In this image you see here, you can see this association between what are integrin interactions and actually growth factor receptor protein expressions. Now, in fact, there's work done by Wei-Kun Zhao of Seidlitz lab here at UCLA, and they actually demonstrated that inhibiting CD44 receptor and integrin subunit alpha V expression, so an integrin knockdown, led to a far greater inhibition following the administration of temozolomide. So this is one way of demonstrating that, yes, in fact, knocking down these cell extracellular matrix interactions can have a beneficial effect in terms of therapy delivery. So beyond what may be an O6 MGMT, there are features in the matrix that contribute to this mounted resistance response that we're only now beginning to see. Now with that, in going over all three systems and obviously offering just snippets into what are vast fields of knowledge, I'm hopeful to have communicated that there's this fact that beyond cancer cells, we are dealing with a cancer system, and it involves extracellular matrix components, which go well beyond the few mentioned in this lecture, the blood-brain barrier, and of course, the presence of these immune cells. Now, while it's beneficial to study these in isolation, each individually, and that's actually what I presented to you mainly within this presentation, it's more important, and this will be done more and more with time, to combine these systems, to combine immune, ECM, these vasculature interactions, to evaluate the effect of mixed systems in vivo. This leads us to the advent of hydrogels and organoids as examples of 3D systems that can be utilized to allow us to mimic and study the combined effects of these immune, vascular, and matrix factors in the mounted resistance response against chemotherapy, or just observe phenotypes, right? Migration, proliferation seen in these grade 4 glioma tissues. Now, these systems allow us to ask even more questions, right? So for example, immune cells also contribute to the deposition of ECM. I mentioned earlier that many of these cells actually deposit their own matrix. Do we have to regulate immune cell matrix deposition as well? Do our effects of controlling these matrix properties actually change the blood-brain barrier functionality? All of these are actually open questions as we continue to explore this GBM system at hand. Moving beyond chemical modifications and therapeutics, there's also this new wave of scientists and engineers in industry emerging that have produced a first of its kind FDA approved device for treating glioblastoma. First of its kind, of course, for glioblastoma. And it's done specifically using electric field. Now, in theory, the company proposes that the electric fields disturb cell division and reduce both migration and proliferation in the GBM cells. And although the trials are limited, they've demonstrated that A 90% compliance rate by patients actually extends survival from 15 months, and that is from the current standard of care, right, with temozolomide resection and applied radiation, to actually 25 months with this new system called Optune. The Optune current device actually emits 1 to 3 volts per centimeter alternating current at about 100 to 300 hertz frequency, of course specified to the patient. And it's supposedly this very optimal solution, and it's done by an electrode cap worn by patients for certain periods of time throughout the day for a certain set of hours. Now, that's not to say that this is any optimal solution, right? 25 months is, again, fantastic, but perhaps tuning current parameters and supplementing device with chemotherapy use, temozolomide or other chemotherapies that are being created, as I mentioned, these EGF receptor inhibitors, vascular endothelial growth factor receptor inhibitors. These may all be used in conjunction with this device and provide an even more substantial response and even more prolonged life in patients. And what remains to be further explored is actually along the lines of using electric fields. These are glioblastoma cells located in the central nervous system, and they contain ion channels. So how does applying electric field actually perturb these ion channels? Can they be used as a way of modulating migration, proliferation, and survival by actually directly affecting channel activation with electric fields? Could this take place of chemotherapies that are actually being currently offered that target proteins or DNAs in the cell? With that said, I would like to return to the definition of glioblastoma multiforme. And I would love to actually accentuate, even with the progress made in our field and our understanding of GBM, of glioblastoma, and improved therapeutics, it remains categorized as the most aggressive, common, and lethal of all primary brain tumors. Now, by improving our understanding of the tumor microenvironment, by adopting this systems thinking approach, we will arrive at better options for patients, better solutions for patients that are heavily affected by this cancer. More than a specialty, I hope you see neuro-oncology as a battle on multiple fronts that requires a systems level approach to designing what will be the future regimen of therapies for our patients. And with that, I'd like to leave you with a message of resilience. Quality embodied by the patients facing glioblastoma, of course, and one that should be embodied by the bevy of physicians, scientists, and engineers developing the future of their care, both now and beyond. Under the current circumstances of George Floyd's murder, this statement is highly applicable. Whether it be 1 or a billion people affected by glioblastoma, whether it be 1 or a billion people affected by police brutality, it's imperative to realize this in no way, shape, or form belittles the significance of the issue at hand. And through resilience, there's always a possibility towards resolve. Thank you for your attention. And should you have any questions or concerns or further interests in the topic itself, please feel free to contact me via email. I wish everyone good health and have a wonderful rest of your day. Thank you.